Today we're all here for the Jones Seminar, and it is my distinct pleasure to introduce you to Dr. Joff Silberg, who is the Stewart Memorial Professor at Rice University. There's a lot of other information on this bio that I'm not going to bore you with, but Joff has an awesome talk prepared for you today. And I have heard that, in fact, a significant fraction of this talk recently got accepted as a Nature paper. So you guys are in for a special treat. And with that, Joff, go ahead.
Thanks, Dan. So first off, thanks to Dan for the invitation to come here today. And it was been really wonderful to have conversations with the students and the faculty. What I wanted to do today is talk a bit about the research we've been doing. I think we sit on the fence between fundamental and applied research. And so where I'm going to go in the talk today is really going to be eventually getting to engineering living sensors. But the path I'm going to take there is really going to go through some fundamental work that we've been doing over the years that led us to realizing we could do some of this fun synthetic biology. And I think I didn't really know what discipline I was in when I started my lab, but I'm pretty sure I'm now a card-carrying synthetic biologist. So that'll be the theme of the talk today. So for me, I find cells amazing because they have an incredible ability to dynamically sense their environment. They have many channels of sensing that they're using in parallel, and they're integrating this information to respond to their environment at the micron scale. So as you look in nature, you find microbes everywhere. So, obviously beautiful coral reefs, soil, rivers, but even in the deep earth, you find microbes, and people that have gone as deep as 4,000 meters find microbes in the deep earth. And these microbes that live in all these environments sense all kinds of things. So they'll sense nutrients, they'll signal each other, they'll sense the physical conditions. And in some ways, they're like a miniature mass spectrometer at the micron scale. Something we've been very interested in is thinking about the diversity of ways that microbes sense things, but trying to leverage these to convert them into information that's easy to access. And so that's really going to be the arc of my talk today. As an example of how much sensing they do, I showed the number of sensors per 100 genes in microbes as you go into this wacky environment of deep earth here. That you see that on average, microbes have 1 out of 100 genes that are for sensing. So on average, they have 50 to 100 sensors per microbe that are dynamically reading out their environment. Now, frequently, to do this sensing, microbes have evolved what are called two-component signal transduction systems. So they have effectively what is a baseball glove on the cell surface that's sensing the molecules. When they sense something, that information is transduced through a phosphocascade to a second biomolecule inside the cell, which binds to DNA and turns on biological production of transcripts and proteins. These are the kind of most common sensing systems in biology, and about 1 to 2% of all microbial genes are represented by these components. So this is how cells are really sensing very diverse things in their environment. And synthetic biologists have been really excited about this ability for a long time, and there are a lot of people that leverage this signal transduction for synthetic biology. So there are people who are building microbes that will go into your gut microbiome and sense when there's dysbiosis. And they'll make a color, or they'll make some type of output that can be detected. And so this is really an approach that's being widely used in synthetic biology as a way of reading out this sensing ability. Now, microbes are really, have an amazing advantage if you were to benchmark them against a mass spec. So I show on the left here my GCMS in the lab. This was about $100,000. It was actually a little expensive. It's about a meter length. You think about microbes, you just grow them. They're micron scale. This thing needs power, pretty intensive. It's got a vacuum. Microbes, you just feed a sugar. They can do their sensing. Service contracts, this is 10% of the cost per year. These are self-healing. The input for mass specs, you got to do a lot of processing before you put the samples in the instruments. Microbes can sense in situ. If you want to sense in a soil in your gut, they can do it at the micron scale in those environments. One of the pluses of mass specs is they generate electrical information, whereas when you think about microbes, mostly what people are getting them to generate are visual outputs, things that can be spectroscopically kind of interrogated. So microbes have a lot of advantages, but one thing that we've been really interested in is, can we convince them to generate an electrical output of the information that they're sensing? So as you look at state-of-the-art biosensors, so synthetic biologists, when I read the literature and look at what they're building, you find that they're really diversifying the sensing capabilities of microbes a lot, but there are some really fundamental limitations of what they're doing. So the first limitation is a time scale challenge. So it's pretty easy to program a microbe, E. coli, or even something that's maybe not domesticated like E. coli, to couple a sensor to a colored output. Doing this in a microbe can be slow. It can take 30 minutes to an hour for the fastest systems to go from sensing an input to generating the output. And so this is because they're using transcriptional regulation in synthetic biology to program this. We're very interested in whether or not you can get to fast responses by using protein level switching or allosteric control of proteins. And I'm going to tell you some stories today about how we've been able to achieve much faster sense and respond time. So there's a major timescale challenge. These are not real-time sensors. Another challenge that people face in synthetic biology is a component challenge. So I just showed you that microbes have all these sensing systems, but it's very slow for us to domesticate those components and use them for synthetic biology and figure out what many of those components are doing in nature. So this is another major challenge that we face right now in synthetic biology. And the third challenge, which is really an area that I've been very interested in, is the output challenge. So as I showed you, microbes are frequently programmed to sense things but generate a colored output or a fluorescent output. But when you start thinking about the environments where most microbes on the planet live, soils and sediments are actually the biggest pools of microbes, but these are very complex settings. So I show kind of a complex feedstock in a reactor. This is, in Houston, we call a river a bayou. Notice how muddy it is. So, not the most optically clear. And soils, where you might want to study and do sensing for agriculture and other applications, are pretty messy environments where it's very hard to measure directly the fluorescence or some of these visual outputs. So my group over the past 10 years has really focused on thinking about new types of outputs for synthetic cells and thinking about how can we program cells but use different outputs that are going to be compatible with environments like wastewater sludge, soil, these bayous that are messy. And so I'm going to talk about one area of the lab today, which is really to focus on how microbes can convert chemical information in the environment into electricity at their cell surface. But we're also very interested in thinking about how can you make rare indicator gases where a microbe might blog about its experience or spy on other microbes and make a rare gas that you can detect in the volatiles that come out of a soil. So we work a lot in this space, and we've also started to build a number of kind of genetic memory devices where we have microbes write information not into their DNA, actually into their RNA. We're taking some different approaches from others. But really what I want to focus on today is my interest in electrical outputs and how we've been thinking about this from a biological perspective. So here's my dream. So this is a dream that I had about 8 years ago. And I never thought we would actually get here. I'm going to show you that we've got some prototypes working. So I wanted to build a microbe that is able to sense some environmental condition. Imagine a chemical, some pollutant that causes microbial blooms, or some toxin that is bad for humans, like an endocrine disruptor. So I would have a sensing element that in real time would sense that chemical and actually couple the sensing of that chemical, like this endocrine disruptor, to electron flow, where those electrons would actually go through the membrane and I could sense them outside of the cell. I really like this application of synthetic biology because I love the idea of taking these synthetic microbes and embedding them in a digital device, like a digital pill that you could take, in part because they're retrievable. So you wouldn't mix this microbe with your microbiome, you'd put it into a device that you could retrieve. So from a safety perspective, it's very appealing to me as a synthetic biologist. But again, the idea is fast sensing where you generate current at the cell surface. So as you think about this, really what I want to do is I need to overcome 3 major challenges. So the first challenge is a protein component challenge. I need literal protein electrical switches. I need biomolecules whose electron transfer is triggered by a binding event to an environmental chemical. And so there aren't many good switches that you can kind of pull off the shelf from nature. So this is something we've had to work on designing, come up with design rules for. The second thing you need to do is when you look at cells, the membrane is actually quite electrically insulated. So you need to be able to get the electrons through the membrane, and I need to somehow plug that into a material. And then, of course, I need a cell that's going to be compatible with my switches, the electrons going through the membrane. I don't show here that there's actually another whole materials challenge, which is how do I encapsulate a cell in a material that's conductive and doesn't kill the cell? And this is actually another challenge we've had to work on, but I don't have time to talk about today, which has been in collaboration with Carolyn Aho Franklin and Rafael Verduzco. So one of the big challenges with getting this to work is really that cells have a lot of biomolecules whose job is to transfer electrons. There are a variety of molecules that have evolved to take an electron from one reaction, hand it to another. They don't do enzymatic catalysis, they just transfer electrons. And there's three different flavors of kind of the low potential electron carriers. There's two types of ferredoxins, and I'm going to use the word ferredoxin a lot today because that's what we've been engineering. And then there's these flavodoxins. If you were to look at the biomolecules in a cell, which this cartoon is meant to illustrate, there are a lot of biomolecules that are catalyzing reactions that need to dump their electrons somewhere. And so they'll dump them to these low potential electron carriers, which will then transfer them to another set of oxidoreductases or biomolecules that catalyze other reactions. And these proteins, these biomolecules, can often take electrons from different reactions. And so I draw this as a network diagram like we understand the flux, the electron fluxome, but actually I draw this as a cartoon. I would love to understand this, but we can't measure that electron flux across the metabolism very easily. So this is a real challenge. We've got a bunch of proteins that transfer electrons. We don't actually know how to control that very well. We've done bioinformatics where we mined 5,000 different microbial genomes and just said, how abundant are these protein electron carriers? The leaves of this evolutionary tree are color-coded by abundance of each of these across eukaryotes, archaea, and bacteria. And what you see is that microbes have a lot of these. So there's actually very complex control over electron flow in archaea. They have a ton of these ferredoxins that are small. As you look at cyanobacteria, you can see the evolution of these 2-iron, 2-sulfur ferredoxins. And you see various pathogens have evolved to low iron environments and use these flavodoxins. And so I just kind of highlight here that there are different organisms that have different lifestyles for transferring electrons. So we kind of have that sense, but we still can't predict the electron flow very easily. The second issue is how do you get electrons across a membrane? So there are some really cool microbes out there that do cool electron transfer that people are discovering. So this is my basset hound. If he was here, he would be howling at you right now. He would be howling because this is his preferred nutrient source that has, some nice sugars in it. He would prefer to eat this, respire those sugars from the peanut butter cookies, and he uses oxygen as a terminal electron acceptor. So we do the same thing. I try not to eat too many peanut butter cookies now that I'm over 50. But Schwannella, which is this cool microbe, it has evolved to respire. It likes lactate, that's its preferred nutrient source, but it actually will deliver electrons through its membrane to ferric iron and reduce that to ferrous iron as a terminal electron acceptor in its metabolism. So it's evolved a way to literally plug into a material. So it can do this on a material, it can do it with extracellular ions. And we actually know a lot about the proteins that get through the membrane, the electrons through the membrane, from the inner membrane to the outer membrane to do this. And in fact, these biomolecules can be used in synthetic biology. So Carolyn Aho Franklin has shown that you can actually port this whole protein system into E. coli, and E. coli will reduce ferric ions. So there's a challenge of, we know that this works, but we don't know how to make this into a sensor in some way. Okay, so how can you study electron transfer? How can you do the engineering, to think about protein electron carriers for synthetic biology? I've just told you the real challenges for these proteins. We don't really know how they plug into donor proteins that give them electrons, how well they plug into acceptor proteins. This is a real challenge. And so I am a biophysicist slash kind of biomolecular engineer by training. I never thought I would love genetics, but I love genetics now. So really what has allowed us to do some fun engineering with these systems is we've started to build cellular selection. So we're able to couple the growth of bacteria to an electron transport pathway. So we are not just putting one protein into a cell and saying, does it function? We are building 3-component synthetic electron transfer pathways. And only if there's electron flow do we see growth. And so whenever you see growth, and I'm going to show you a lot of data, basically what that means is the biomolecule is conducting electrons through this pathway. And so this assay that we built has been incredibly powerful for various types of studies. So the first thing I want to show you is just kind of the kind of data I'm going to walk you through as I go through some different vignettes today. So imagine you have a cell, and that cell has these proteins. Now, we build this at the genetic level, so these cartoons are meant to be our genetic circuits. This is what we're designing. This is what we're putting in cells. The electron donor, which is FNR, is always made. The acceptor, sulfite reductase, is always made. The cells get fed sulfite, and the only way they can live is if they can convert this sulfite to sulfide, because otherwise they can't make critical building blocks. So if you make the donor and the acceptor, there's no growth because there's no current, because these can't transfer electrons to one another and support growth. What was really cool when we started to work with this assay is that when you turn on the production of the ferredoxin electron carrier, what you see is how much you turn it on. So this concentration is kind of how much we're producing here. We're inducing its production. That you see it turn on, and in a dose-dependent manner, you see more growth, which you would expect if this is the rate-limiting step for electron transfer. And so one thing that's really cool is when you start looking at data this way, we can start abstracting biological components much like the electrical engineers do. So you can start thinking about transfer functions for like how much of this component I make, how much electron flow is there. So kind of what concentration of it do I need to get half maximal current? And so this is something that allows these assays to look at many different biomolecules that transfer electrons in parallel. So we can use this growth as a very high throughput design assay, or even to ask fundamental questions about how different biomolecules differ in their ability to couple electron transfer between different donors and acceptors. So I wanted to tell 3 vignettes today. All of these have leveraged this pathway. So this genetic tool has been incredibly powerful for us. The first vignette I want to tell you is really about evolution of these protein electron carriers. The second is going to be a very engineering focused one where I'm going to talk about how we built electrical switches. And finally, I'm going to get to kind of an electron transfer pathway we built for sensing. So these electron carriers, as I show on the right over here in modern cells, they bind iron and sulfur cofactors. So these are the cofactors that are actually holding on to the electrons, giving the electrons to the partner. As you look in nature today, these proteins actually have a twofold symmetry of axis. So they look like they evolved from a smaller protein that duplicated and then diversified its sequence. People think that these are one of the earliest proteins to have evolved, and there are theories that these actually evolved from an ancestor that was symmetrical, or maybe even a peptide that was smaller, as you think about evolution at kind of these deep sea thermal vent kind of environments. So we've been using this assay to ask the question, can we make proteins that might look like these ancestral electron carriers, and using our assay to think about that. So the way this works is we went in and took a large number of these protein electron carrier sequences, right? There's thousands, tens of thousands in nature. So we took a large number of those sequences and we figured out what would be a consensus sequence and designed 2 of these. And all of this work was with Vikas Nanda at Rutgers. As you design these, they were asymmetric in our consensus design. Then what we did is we said, okay, let's take 1/2 and then duplicate that so it is what we're calling symmetrical, but really it's just duplicated, 2 peptides duplicated. We then built those and asked the question, in a test tube, will they bind an iron-sulfur cluster? And then asked the question, after we built them, can we put them in our assay and can they support a cellular reaction? So we're really asking the question, if you built a putative ancestral protein, can we see that it functions in a cell? And when we did this, we were surprised to see that when we built consensus sequences that had this symmetry here, they all bound their metallocluster. They had midpoint potentials that were very similar to the parental protein. So the midpoint potential is really a way of thinking about how much it wants to hold on to or give an electron to a partner protein. We had two types of designs. One of them was a consensus design seeded from a really large pool of ferredoxins. And then this is actually the first one that was done, which was a smaller kind of bioinformatic pool. And in all cases, they bound metalloclusters and had low potentials, much like natural modern ferredoxins. What was fun was when we went and put them in our assay, we found that some of them could actually complement growth. So remember, growth for our assay is whether or not it's transferring electrons from a donor to an acceptor protein. And so we found that some of these biomolecules could not support growth, but some could support growth. And so this is really exciting for us because it suggested that these proteins did evolve from symmetrical ancestral proteins. More recently, what we've been doing is we've been asking the question, can you take this protein that's symmetrical, break it into smaller bits, and try to go farther back in evolution? And that's what we've done more recently. So we've made actually these 28 amino acid peptides. They have a number of cysteines, just like the full-length proteins, but they basically cannot coordinate an iron-sulfur cluster by themselves. You actually need two peptides to come together to get the metalloclusters to be coordinated. Vikas Nanda has shown that in vitro, these will all coordinate iron-sulfur clusters. And we've also taken these into cells and asked the question, do these peptides, so these are like half of one of these electron carriers, can they support electron transfer? And again, remember, when you see growth, which is this optical density here, you see that there's an increase in growth when you express these proteins. What we do see is that they're exquisitely sensitive to oxygen compared to even the full-length proteins when we do this in E. coli. So they're even more sensitive to oxygen than kind of the full-length proteins, but they will also function in cells. And so what this really shows is that we can use this assay to test fundamental hypotheses about the evolution of these biomolecules, which were thought to be critical to ancient metabolisms. Now, one thing you could ask is, well, is this, yeah, please.
Sure. Can you read from the answer question? No.
I'm open to questions anytime.
I'm just curious, how did you decide various proteins? And even before when you were doing this metric,
yeah. So this is based on evolutionary conjecture. So if you look at these proteins, and I've tried to show it as a cartoon, this bit here that's checkered aligns with this bit that's checkered. And so the idea is that in evolution, what happened was you had one checkered bit, there was a gene duplication, and it became fused, and that's what we see today. And so the yellow bit is kind of a small linker, 3 amino acids, and we chose to cleave it right before that. Please do stop me with questions. So more recently, we've kind of asked the question, might these exist in nature? Have people not found them? And so we've actually gone into about 7,000 genomes and looked for ferredoxin-like peptides below the size cutoff that's typically used for the native proteins. So the natural clostridial ferredoxins are about 56 amino acids, which is a really small protein. So when people do bioinformatics, they actually will use size cutoffs so they don't get lots of smaller things. So we've gone looking for the motifs that bind these iron-sulfur clusters, and we've actually found many smaller peptides. In fact, we found some that are very similar in length to the ones that we found. They're a little bit different in sequence, but these types of peptides may actually be coordinating iron-sulfur clusters in living cells. Vikas has actually made some of these after we found them, and in vitro, they will bind iron-sulfur clusters. So we don't know what they do in cells, but it may be that nature still uses some of these smaller peptides for different functions. So really what I wanted to illustrate with this is this idea that these cellular assays are very powerful for asking questions about a very hard to study reaction, which is electron transfer. And really, as we started to work with this assay, we started to think more like engineers of what could we do with it? So as we were looking at electron transfer, the reason we got into it was really to think about the fundamentals of control of electron transfer as they relate to metabolic flux and kind of partitioning of electrons across different reactions. But as we started to think about having an assay, I did my postdoc with Francis Arnold, so I couldn't help but think, what could we engineer with this now that we've got a high throughput assay? And so Josh Atkinson, who's a wonderful graduate student in the lab, he and I discussed whether or not we could build protein switches out of these paradoxes. So remember, these proteins, their job is to grab an electron from one donor, give it to another protein. But you could imagine thinking about them much like we think about a light switch. Could you engineer them so they are conditional on when they're going to do that electron transfer reaction? And so our idea was, could we somehow engineer them so that they were fused to something that binds a chemical, and that chemical binding caused a conformational change that affects electron transfer? Now, this was one of those projects, you have high-risk projects, and we talked about this. We didn't think it was going to work because we're like, boy, if you start messing with this protein, is it still going to bind the metals? And if you tweak it a little, maybe there'll be stability issues or you'll break it in some other way. But it's one of those experiments that was worth giving a shot. And so it ended up, you'll see, being a very fun outcome. So as we thought about this selection, we started to think about how could you build a protein switch? And really, you need a good off state to build a switch. And so our first idea was, can we figure out how to build a good off state in this biomolecule by breaking it into pieces? So our idea was maybe we can break it into pieces and then understand how to bring those pieces back together in a way that's dependent on the environmental conditions. So the idea here is going to be we're going to engineer some variants and we're going to put them in the cells and use the growth as a readout of, do they still transfer electrons? We did this, we did two things to gain this. So the first one was we chose a protein that is very thermostable. So as you look at people who do protein design, there's very good evidence that when proteins are more stable, so example, if you take a protein from a thermal vent and you bring it into this room, so it evolved at 90 degrees Celsius, but we're bringing it into this room that's, like 23 degrees Celsius. That protein is going to be more robust to tolerating mutational change and still fold into its beautiful structure. So we took a very thermostable protein, so we were hoping it would still kind of function. And then we took a lot of sequence data from many family members and kind of looked at family sequence conservation. And we knew we shouldn't break it too close to the cysteines that bind the irons because we would destroy the cofactor. But we made 4 different variants that were, not quite at kind of peak divergence, but maybe intermediate divergence. And we do this a little bit more rational now, but this is the first time we've done it. What does this look like? So these biomolecules, so I show this ferredoxin, it's got an iron sulfur cluster right here. So the first variant we break, and it splits off just a beta sheet. And so most of the bits are on here. So the bits on that big chunk are the things that bind the cofactor, bind the donor protein. So those purple residues are the things that bind to the thing that give it electrons. And then the green are the residues that bind to the acceptor. So those purple and green are what it needs to interact with. So we're pretty far from all the important bits of the protein, we think. We then split it in different spots. So this spot, for example, breaks it so half of the cysteines that bind this iron sulfur cluster are on one bit and on the other. So we did some more dramatic things to it. And when we built 4 of these, and so this was kind of the preliminary data for this project, Josh came in, showed me the data, and he's like, look, here's the wild type, the full-length protein. It supports electron transfer. Here's the negative control. So this has one mutation that breaks iron binding. It doesn't grow. They're all dead. And he was like, I'm not sure this is going to work. And I was really excited. I'm like, this is great. We've broken these really well. All we need now is a way to kind of help them come back together. So we've got a really good off state when we break this protein into pieces. And one of the things, that I'd seen in the literature and he and I had talked about is chemical complementation of protein-protein interactions. So there are, first thing before that. The next thing we did is we asked the question, if we fuse peptides that drive the association of these fragments, do the fragments now function? And so what we found here is that if you just have the fragments, they cannot support growth, as I show here. But as soon as you fuse it to proteins that drive the interaction, you get electron transfer again and growth. And we saw that for all of them. So all of them had conditional activity for electron transfer. And so this was really exciting because it showed that we could find tricks to kind of turn on the electron transfer and switch it on. So what I really want to do is I want to sense chemicals in the environment. So I want these electron transfer proteins to sense a metabolite or some chemical that's maybe toxic in the environment and control their electron flow that way. So the next thing we did is we took the little blue protein fragments, this is the electron carrier that's been split in half, and we fused it to proteins that don't interact unless they see this chemical rapamycin. And asked the question, can we make a conditional protein-protein interaction that is sensing a small molecule that you provide in the environment of these cultures? And again, remember, no growth is no electron transfer, growth is electron transfer. The negative control is just a full-length mutant ferredoxin that can't bind a metallocluster, no growth. Wild type you see up here has consistent growth. And this split protein, when you add the chemical, you see electron transfer or growth that is dose dependent. So this now shows that as we increase the concentration of this chemical, we can see the electron transfer switching on. So this was kind of our first evidence that we can now use a small molecule to turn on electron transfer in these proteins. Now, of course, I want to know mechanism. I think I had some conversations with a number of people today. You know, you do these experiments, and this is a cellular assay, so we can generate quick data. And I'm drawing cartoons like they're, things are functioning this way. But I really want to purify biomolecules, know what's happening. And split proteins are awful to purify. They get degraded, they're very hard to isolate in high yields. And so the first thing we did before doing that was we thought about some alternative designs. So we were asking the question, could this be a modular design strategy, and could we simplify it a little? So instead of having those two fragments on two different things that bring them together, could we have the two fragments fused to the end of a protein that's known to undergo a conformational change? So it has a big open state, the fragments are apart, it's got a closed state upon ligand binding, the fragments are close. We'd read some really cool work out of Weiman Zhao's lab where there'd been some work with GFP biosensors where they took the estrogen ligand binding domains. This is the human estrogen ligand binding domain. There are a lot of structures known for this. So there is a structure of the open conformation with no ligand. The N and C termini are 60 angstroms apart. There's then a structure with agonists, so that would be like estrogen. They're 33 angstroms apart, so they're a little bit closer. And if you add an antagonist, so bisphenol, environmental pollutants, and endocrine disruptor, that would be an antagonist, they get even closer. So we thought this would be a really cool test case for fusing our ferredoxin fragments to a ligand binding domain. I draw this cartoon as like a dream of how it works. I actually don't know the mechanism and what it looks like, but it's a nice way to conceptualize it. The open state has the fragments far apart. The closed state, upon adding a ligand, has them together. And again, now I show growth, and this is kind of a high level, high concentration of various chemicals. When we built this design, if you add agonists, that is the agonists that put this protein ligand binding domain in this conformation in the middle, we see that none of them complement growth more than cells that don't express this protein. However, when we add very different estrogen disruptor antagonists, ranging from hydroxytamoxifen to bisphenol, we see significant growth here. And we see it in a dose-dependent way in all cases. So we have a lot more data where we've kind of looked at this, but what this now shows is we can make a single biomolecule that's acting as an electrical switch. So this protein, when it doesn't have ligand, is a very poor protein electron carrier. But when it binds the ligand, it can transfer electrons very effectively, is what this data shows. Now, we couldn't help but try to go in and do a little bit of mechanistic work. So as we did this work, for anybody that works on iron sulfur proteins, these can be very complex to build on proteins. So there's elaborate machinery in cells that evolve to build these clusters. And sometimes they're hard to express in non-native hosts. Sometimes you make a mutation and cells can't hold on to those clusters. And so we really wanted to understand, if we purified our switch, does it have a cluster bound before it switches on? So you could imagine a couple cases. Cells are making this thing and it gets degraded or it's not functional. And when a ligand is around, then it puts the cofactor on. That could be a very slow process. But if you want a fast switch, you want a biomolecule that's poised to go as soon as diffusion-limited binding occurs in a conformational change. So what we really wanted is to see, does this thing already have the cofactor before it switches to the on state? And so what I show here is kind of the absorb. So the first thing is we expressed it in cells, we put it on a column, and the column turned brown with oxygen around. And I was like, oh my God, this is exciting because this thing probably has an iron sulfur cluster. We went and compared the visible and circular dichroism signals for the wild type protein as well as our fragmented one fused to the ligand binding domain. And you see they have similar peaks that are consistent with having an iron sulfur cluster of a similar type. We also went and measured in collaboration with Sean Elliott at BU, because it proved a little finicky, the midpoint potentials. And so in the presence and absence of one of these endocrine disruptors, we see no change in the midpoint potential of the native protein. We see a small change in the midpoint potential of our switch. We don't think that's actually what's gating it. We think it might be the binding interactions with the partners, given that this is pretty small relative to the midpoint potentials of the donor and the acceptor. But what this showed and what made me really excited was this protein is poised for fast switching, right? It's got a cluster, it's just waiting for a ligand to bind, and then it can transfer electrons. And so this was really exciting for us. And, I think Josh was able to publish this. So, I was so excited when Josh put this together. It was a beautiful thesis, but he wasn't satisfied enough. And so the last thing yet I want to tell you about is two things. What happens when you're not satisfied enough with your results? And the second one is why you should go to a conference. So it ended up, Josh went to the Synthetic Biology, Evolution, Engineering, and Design Conference. And he had a poster on that story I just told you, which is really cool. Lots of people came up. Right next to him was another researcher, Moshe Baruch, who was in Caroline Aho Franklin's lab. Also studies electron transfer. I didn't really know Caroline. Josh didn't know anybody in the group. And what I'm going to show you is what happened when Josh saw Moshe's poster and vice versa, thinking about what's possible with synthetic biology. And so what I'm going to show you is really a story where we can now use those switches to regulate extracellular electron transfer. So our switches regulate the electron flow coming out of the membrane. And this is a 6-year project, that we are so excited. This just got accepted last week in Nature. And I have to note that this is a true 50-50 collaboration between my lab, Caroline Aho Franklin's lab. Neither of us could have done this alone. It was a true 50-50 collaboration by both of our grad students. Josh, both doctors now, Josh Atkinson and Lin Su. And Shu Wang actually was really instrumental for us encapsulating our microbes and materials to get our noise down and our signal up. So there was no way we could have done this without an interdisciplinary team. So two things. So what did we bring together, and why should you go to conferences? So Josh had a poster on our lab's discovery of fast electrical switches. Moshe had a poster on putting proteins into E. coli that wire electron flow through the membrane. So Moshe had a poster on these MTR proteins that are a literal wire through the membrane of gram-negatives like Schwanella. And they allow these microbes to deliver electrons from their cytosolic metabolism to extracellular metals or even an electrode. And so the idea was to build a synthetic electron transfer pathway. So this is all proteins. We're not building DNA kind of transcriptional circuits here. This is make a bunch of proteins and try to build regulation into those proteins for sensing. There are 8 components. So we start with an FNR that's going to oxidize NADPH. Those electrons go to our ferredoxin, which I've told you a lot about so far. That then goes to a sulfite reductase, which takes sulfite to sulfide through a 6-electron reaction. That then goes to a sulfide quinone reductase that oxidizes the sulfide and reduces quinone, which then can go out through the membrane through that Schoenella pathway. So all of these we are engineering in E. coli. So every one of these is non-native. They're from 5 different organisms, but that's not enough. Then we're putting our engineered switch into this and trying to get this all to work. And so I will never tell a student to do this again. We're now building much simpler stuff because we learned a lot from the problems that we had. But to give you a sense of what we're trying to do here, we want the switch to sense chemical information in the environment, convert that to biochemical information in real time. We then want the pathway to transduce that biochemical information into electronic information. We want to read that out with the digital device. So really it's trying to convert these different types of information using the living microbes. So this is now the nitty-gritty of the synthetic biology. So these are all DNA circuits that we had to build. There are 12.5 genes that we needed in these plasmids. We're actually missing a couple of the genes because we had to fix the mutant strain we were working in. We didn't realize there were some broken things. It's a lot of genes. We got a lot of moving parts here that we need to optimize. The other thing is, as you look at the proteins that get made, these proteins, there's 8 of them, they have 29 cofactors. So there's a lot of extra stuff you have to build on those proteins to get them to function. And we actually express some machinery that helps build some of these things in E. coli. So it's a pretty elaborate pathway. We think it's the hardest, kind of the most complex synthetic electron transfer pathway that's been built to date. That's not just porting it from one organism to another, but kind of building it a priori. So as you build this pathway, and just kind of walking you through it at the top, this bit is what we've been working on. So we are working on a donor to our ferredox and to the acceptor, the sulfite reductase. Caroline was working on the red bit, and actually Josh's idea was if we could just couple them. So when he was at this conference, I'm getting texts of like, you got to see this stuff. We need a coupling module. How could we do that? And so we need to figure out, does our stuff work in cells that has Caroline's stuff and vice versa? And can we get some coupling module to work in E. coli that'll wire this all together? So the first experiments that we did were to take our proteins and express them in a strain that has Caroline's proteins and ask the question, do we see growth? So we need a very special strain for our pathway. We use a very special E. coli. It's been mutated. It's insulated in ways that most E. coli strains aren't, so the electrons go where we want. So we've got to actually put Caroline's proteins into our strain. So the blue line shows the kind of growth data that I showed you early on. That's kind of our validation of our assay. And then what we did is we expressed all of these proteins in that same strain and asked the question, does our assay still function? And that's the red here. And you see that our pathway still works even when there's extra burdens on the cells to make these multi-heme cytochromes in the membrane. So it looks like our module works if you add Caroline's proteins into our strain. We then ask the question, does Caroline's proteins work when we add our modules into that and you're monitoring extracellular electron transfer? So this is now current that's being measured in a bioreactor as a function of time. You notice that E. coli makes some current at the potential we're standing at. So you're seeing, this is actually probably riboflavin that you're seeing here. And you can see when you express these proteins in our strain that you actually see current above kind of E. coli's normal current. So her proteins work in our strain. So this module works, this module works. The third module is the coupling module. So we took cells, expressed different sulfide quinone reductases, added some sulfide. We have a sulfide electrode. If you don't express any sulfide quinone reductase, sulfide stays around. Coli doesn't do anything to it. But if you express different variants with different affinities for the substrate, you see different rates of consumption. So we took the one that was fastest at consuming the sulfide so we could couple these tightly. And then we put it all together. So now I'm going to show you the data, first from putting the whole pathway together. So the blue line here represents the whole pathway, except there is a single amino acid mutation there that breaks the cluster. So basically we break the wire here with the blue trace. So this is current as a function of time as we add in thiosulfate, which is required for current through this pathway. As we dose in thiosulfate, if it's the wild type pathway, which is in red, you see dose-dependent current. So this is like year 5. Like, we got it all built in 4 years, we tuned it all, it took us a long time to get this to work because we actually had a material challenge. So we actually had to encapsulate our cells in conductive materials with an electrode, which is how these are all being done, these experiments. What was really exciting was when we finally got to put our switch into this. So now this is the full pathway. We take this engineered switch, substitute it for this ferredoxin, and we have different reactors. One reactor where we add an endocrine disruptor, hydroxytamoxifen, and the other where we add DMSO. So DMSO is what we use to solubilize the hydroxytamoxifen. And what you see is when we add the chemicals, nearly in real time, you see differences in trajectories of the current. Now, you don't actually see a significant difference in current until you're about 3 minutes, 3 to 4 minutes in this experiment, but it shows that we're getting real-time sensing of the chemicals. One thing I should note is if you do some kind of fixed law type of calculations to ask the question, how long should it take the chemical to get into the little hockey puck, the alginate kind of hockey puck that we have our cells in, it's about 2 to 4 minutes. So we actually think the rate for seeing the time delay for getting a significant signal is really just the chemical getting into the material, not the cells having to make something. What's particularly exciting, and this is kind of the last data slide I wanted to show, is what we've been able to do since then. So with these cells that are encapsulated in materials, we've now done these experiments in multiple messy bayou samples, as well as Galveston Bay marine water. And we've added the chemical in as well as DMSO, and you can see again, you're getting near real-time signals from all of these samples. So encapsulating the cells and materials are actually probably an advantage for working in complex environmental settings. But really what we've got and what we're so excited about is a real-time method for detecting chemicals in the environment and converting that into electrical information. And so this is really where we're at today. We're thinking about a lot more simple designs. And really, I just want to give you a few slides for how we're looking forward. So as we're looking forward, I should note, we're recruiting. I'll show you 3 slides why we're recruiting. So the first is that the DoD has been very kind to us. We have a team project now where we're trying to massively diversify the sensing. So can you come up with some modular designs to make families of electrical sensors and also sense on the outside of cells? But we're also working to actuate by turning on current and coming up with some tricks to do that as well. So could you build a device that senses a chemical like the estrogen antagonist and then electrically turn on cells that actuate and degrade that? So we're working to do this, and this is a big team of researchers. We're the protein design side. We're also, we were just, notice the date here, it's been a good month. We got good news from NSF that they want to support another grant, which is trying to build electroactive microbes whose current is dependent on kind of macromolecules that you might find in environmental samples like wastewater in Houston. Since this has been so good for tracking things like SARS, and in fact, monkeypox people are able to see in the wastewater as well. And you know, we're really trying to think a little bit more creatively about biology and electron transfer. So maybe a third project, and again, it's been a really good year. I never thought I would have this good of a year. NSF has funded us to start thinking about building not permanent memory in cells, but actually trying to figure out how to use this current getting across the cell membrane to read out information that's stored in DNA and RNA. So we're trying to actually build memory in cells that isn't just like write info in the DNA, it's there forever. We're trying to write information into chemical mediators, into RNA that could be around the cell and you could read out at different times by sweeping the potential. And so we're trying to build kind of biological RAM memory right now. Now, I do want to note that everything that we do is collaborative. So nothing in this project, or actually any project in my lab, can be achieved without interdisciplinary teamwork. And I think it's been really cool to be here today and really learn about Dartmouth and kind of the lack of departmental boundaries, seeing grad students from very different training programs together, because I think this has been so critical to all of the scholarship that I do. And actually, I didn't start my career this way. It took me till being an associate professor to realize I should be doing this kind of work, not sitting in my silo. I want to note that all of the last story was a 50-50 collaboration with Carolyn Aho Franklin. And of course, acknowledge different funding sources, and thank you for your attention.